sidebar. So I'll admit that today I'm sort of stretching the definition of developmental neurobiology a little bit, but I really wanted to take this opportunity to present a currently unpublished story in the lab, but we're sort of trying to sort of finish up a publication at the moment. Really trying to understand how remyelination failure impacts neuronal health. And just to give credit where credit's due up front, I really want to highlight that almost all the work I'll be showing you today is the work of a very talented postdoc in the lab, Greg Duncan, a man so enamored with biology that he dissects his Easter eggs, along with Samantha Ingram, a research assistant in the lab. So the interest in my lab is really anything to do with CNS myelination. And as hopefully you're all aware, myelin is sort of a fatty substance in the brain and spinal cord that's formed when an oligodendrocyte wraps its processes round and round neuronal axons, extrudes for cytoplasm, leaving behind these multilaminar arrays of membrane. And it's this sort of very insulative stacks of membrane that allow for the process of saltatory conduction or rapid conduction along axons. Now, the process of myelination is sort of predominantly an aspect of postnatal development. So it occurs largely in the first few decades of life in humans or the first few weeks of life in mice. But I kind of want to highlight that this is really probably best thought of as an ongoing developmental process that occurs right throughout life. And, you know, Kayleen Young's previous work in using genetic fate mapping really very elegantly showed that this is a process that occurs on, you know, throughout life as OPCs will differentiate into oligodendrocytes. So, even though both the developing and the adult brain contain a sort of a resident pool of these OPCs that Kayleen so sort of nicely presented before, that are capable of differentiating, this process can still go awry. And this is sort of really very sort of powerfully shown in human diseases such as multiple sclerosis, where you get a sort of an inflammatory immune attack that will leave areas of axons devoid of their myelin. Now, and in many cases, this myelin can repair, and this is illustrated in this brain here from a person with multiple sclerosis where the myelin's been stained in blue. And you can see these red arrows indicating sort of areas of demyelination that's partially repaired or remyelinated. The bad news is that many other lesions, however, such as the ones indicated by these green arrows, will remain chronically demyelinated, perhaps for decades. And this has several consequences. First, it causes an ongoing block of conduction along these chronically demyelinated regions of axons. But in addition, it's also thought to leave both the axons and the neurons vulnerable to degeneration. And it's partly because of this, but there's really been sort of a very large sort of focus in the area of really trying to promote remyelination or myelin repair. And it's really proposed that this will do two things. One is to get functional recovery of the conduction along demyelinated axons. But in addition, the idea is that remyelination should be neuroprotective, preventing this axonal and neuronal loss. I think it's sort of notable, though, that, you know, in spite of this assumption in the field, there isn't actually a great deal of hard experimental evidence that remyelination is directly neuroprotective. And I think this is partly because the existing mouse models of demyelination, unlike here, humans tend to remyelinate very efficiently, usually within a few weeks. So there's no great mouse model of the chronic demyelination which you often see in humans. So a number of years ago, my lab sort of really started wondering whether we might be able to capitalize on some of our previous interest on sort of the genetic regulation of myelination to try and sort of develop different mouse models for this. And this work really grew out of my previous interest in a gene now known as myelin regulatory factor, or MYRF. And MYRF really first came to our attention many years ago as a gene that was expressed within the brain very exclusively by postmitotic oligodendrocytes. And it really switched on very early during oligodendrocyte differentiation. And a lot of work from, you know, a number of labs over the years has shown that this gene encodes a very unusual membrane-associated transcription factor that actually initially is synthesized as a transmembrane protein in the endoplasmic reticulum, and then undergoes this trimerization and an autoproteolytic cleavage. And you get a bit of a sense for this in this single oligodendrocyte shown here. So you can see the N-terminal cleavage product of the MYRF protein in the nucleus, whereas the C-terminal component of the protein remains associated with the endoplasmic reticulum. And, both us and a number of other labs are still really trying to understand, why this protein undergoes this fairly convoluted process. What's really very, or comparatively well understood about this protein, though, is that it's absolutely critical for oligodendrocyte differentiation and CNS myelination. And I think this is probably most vividly shown with this image here in the spinal cord of a control mouse. And you can see it's pretty much full of myelin basic protein staining, staining the myelin. Whereas the mouse in which the MYRF gene is being conditionally ablated from the oligodendrocyte lineage is completely devoid of CNS myelination. And in contrast, the peripheral myelination is just fine. So I really want to highlight that this gene is absolutely critical for the formation of myelin in the CNS. Now, a number of years ago, while the lab was still in Melbourne, Matthias Koning, a graduate student, started looking at whether MYRF was only required for the formation of myelin, or whether it was also required for the maintenance. And to look at this, he made use of a PLP-CRE driver, which is a tamoxifen-inducible CRE, which is expressed within the post-mitotic oligodendrocytes. And what he found is if you let mice age normally before giving them tamoxifen at 8 to 10 weeks of age, by which time myelination is largely established, what you'll get is a very slow demyelination as the myelin sort of breaks down and the mature oligodendrocytes undergo apoptosis. And this occurs over about a 10-week period. And then over a subsequent approximately 10-week period, the progenitor cells, the OPCs that Kayleen has discussed, will proliferate, differentiate, and remyelinate through the generation of new oligodendrocytes. So, there seemed to be quite an interesting sort of genetic model of demyelination and remyelination. And for a long time, we sort of didn't take this observation much further until Greg came into the lab and started wondering whether we might be able to tweak this genetic system in order to block for remyelination and give us a sort of a mouse model of chronic demyelination. So the way Greg did this was to, instead of using the PLP-CRE driver, as he used a SOX10-CRE driver. So SOX10 is a transcription factor that's expressed right throughout the oligodendrocyte lineage, both in the mature oligodendrocytes, but also in the oligodendrocyte progenitor cells. So the idea is that the ablation of MYRF in the mature oligodendrocytes would once again give the demyelination. But in addition, and probably more importantly, the ablation within the progenitor cell population should prevent the differentiation and generation of new myelin. And these two lines I'll refer to for the rest of the talk is MYRF delta SOX10 for these non-remyelinating mice, and MYRF delta PLP for the remyelinating mice. So what these lines basically look like in practice is we usually wait till these mice are about 8 to 10 weeks postnatal, so largely myelinated. And you get a bit of a sense for this in this black gold stained brain here, taken just at the time of tamoxifen induction with the myelin stained in black. And if we take these MYRF PLP line, give them tamoxifen, they'll develop this sort of a development of the paralysis over about a 10-week period. So this is a partial hind limb paralysis and also quite a severe ataxia. And this corresponds very nicely to a loss of most of the myelin throughout the central nervous system. And you can imagine it's probably loss of myelin in the cerebellum, for instance, that results in this ataxia. And these mice will then sort of partially recover over sort of the next 10 weeks. And once again, this correlates very nicely with the formation of new myelin, which you can get a sense of in this black gold stained brain here. Now, if we repeat this experiment with the SOX10 line, what you can see is that the onset of motor symptoms is quite sort of similar to the PLP line, but they develop a much more severe disease. In fact, most of these mice form pretty much complete hind limb paralysis. They show sort of no real signs of recovery. And unfortunately, they usually develop seizures. So we've found we typically have to euthanize them at around 12 weeks post-tamoxifen. But it's a really much more sort of aggressive disease course for these than the mice that are capable of remyelinating. Now, I also want to sort of shift gears a little bit here and just point out that while both of these mouse lines have demyelination throughout the entire central nervous system, everything I'm going to be presenting you from now on is really focusing on the retina. And we've decided to look at the retina as sort of a simplified component of the nervous system for several reasons. One, obviously, it's a somewhat simplified or accessible circuit compared to much of the brain. But it also has a population of neurons for retinal ganglion cells, which within their optic nerve component of the projections are pretty much fully myelinated. So it's a very heavily myelinated population. And we also know that it's a clinically relevant neuronal population. So in fact, thinning of the retinal nerve fiber layer, which contains the axons of these retinal ganglion cells within the retina, is actually sort of one of the standard clinical measures of axonal loss in multiple sclerosis. So everything I'll be showing you from now on is in the visual system. So if we look at the optic nerve, we have here a controlled animal. So this is a pre-negative animal, 10 weeks post-tamoxifen. All the axons within the optic nerve are being captured in cross-section here. And you can see in this control animal, pretty much every axon is ensheathed with this layer of myelin, which is visible in these electron micrographs. In contrast, if we take one of these MYRF delta PLP mice at 10 weeks post-tamoxifen, you can see that there's a very substantial reduction in myelin. And in addition, many of the myelin sheets that are present are quite thin. And this is really a cardinal feature of remyelination, which starts off quite thin. And then if we give these mice an additional 10 weeks, you can see that there's even more substantial remyelination. And if we quantify this over the sort of 20 weeks post-tamoxifen, about 70% of the axons in the optic nerve have undergone demyelination and then remyelination. As best we can tell, the myelination never gets back up to a healthy baseline. Now, this can be sort of contrasted with the MYRF delta SOX10 mouse line, which again starts off fully myelinated. But if we look at them at 10 weeks post-tamoxifen, there's really a complete loss of the myelin in the optic nerve. And if we quantify this, we've gone from about 95% myelinated axons in any given cross-section for the controls, down to maybe 2 or 3% of the axons myelinated in any given cross-section in the delta SOX10 mice. And what you can also see is there's sort of none of these thinly myelinated axons, really suggesting we've completely blocked remyelination with this genetic strategy. Okay, so if we block remyelination, what's the consequence on the neurons? So to look at this, we've looked at retinal flat mounts within these animals, staining them with RBPNS, which is a marker for the retinal ganglion cells, and cleave caspase 3 to catch actively apoptotic neurons. And if we do this within a control mouse, you can see the RGCs here, and you basically never see an apoptotic cell. In fact, what we're able to do is sort of flatten out the entire retina, and we can actually quantify basically every dying RGC over the entire retina. And you can see in a healthy mouse, there's really only a handful. As a positive control for cell death, we've got here the retina from a mouse 3 days after an optic nerve crush, and you can see there's a sea of dying RGCs, which we would expect. But encouragingly, we also see examples of apoptotic RGCs within the retina of these MYRF delta SOX10 mice that are unable to remyelinate. So if we go across and quantify the density of dying RGCs in the remyelinating line versus the non-remyelinating line, you can pretty much see right off the bat that there's really only a significant increase in RGC death in the non-remyelinating mouse line. And we see this at both week 10 and week 12 post-tamoxifen. In contrast, the mice that are capable of remyelinating show very little RGC loss. And I'm not going to go into this data today, but we see something very similar if we quantify axons within the the optic nerve. So we also see a loss of axons within the non-remyelinating mouse line, but not the myelinating or remyelinating mouse line. So I think this is kind of exciting because it really shows, very clear genetic evidence that, firstly, that remyelination is neuroprotective. I think we've shown that genetic ablation of MYRF in mature oligodendrocytes results in a delayed CNS-specific demyelination. And I think the additional neuronal death we see in the non-remyelinating MYRF SOX10 line really gives us a unique model to sort of start looking at the mechanisms underlying neurodegeneration in response to remyelination failure. So in the last few minutes of my talk, what I'd like to talk to to you about is again, unpublished data where we're starting to look at one of the candidate pathways that we think might be driving neuronal loss in response to remyelination failure. So when we did RNA-seq analysis of the retinas from these chronically demyelinated mice, one thing that really struck us is we saw an upregulation of a number of genes that were known to be regulated by the c-Jun transcription factor. And the reason this was striking is the c-Jun transcription factor is a member of the pathway that is known to be very highly involved in axonal stress and cell death in response to a number of injuries. And this is the DLK-JNK c-Jun pathway. So DLK, or dual leucine kinase, zipper kinase, is a protein that senses a variety of axonal stresses, and when activated, will phosphorylate MKK4 and 7, which will in turn phosphorylate JNK, which finally will phosphorylate c-Jun. And depending on the neuronal type, this can have a range of transcriptional responses. In CNS neurons, this usually initiates a programmed cell death pathway. However, in the peripheral nervous system, it can be associated with activation of a regeneration pathway. So we sort of wondered whether this pathway might also be activating cell death in response to the demyelination. So we first looked to see whether we could see any evidence of activation of c-Jun within the retinal flat mounts. So here we've got phosphorylated c-Jun stained in green and neurons in this case stained in magenta. And I think what you can appreciate if you look at the control panel here of a healthy mouse is you can basically never see activation of a c-Jun transcription factor within the RGCs. Again, as a positive control for injury, we've got a retina here 3 days after optic nerve crush, and you can see there's a sea of activated c-Jun in the retinal ganglion cells, as we would expect. If we look at the remyelinating PLP mouse line, you can see only very occasional examples of activated c-Jun within the retinal ganglion cells. And this is sort of greatly increased in the non-remyelinating delta SOX10 line. One thing I sort of want to highlight is that even though the density of these phosphorylated c-Jun positive RGCs is much lower than, for instance, in optic nerve crush, the actual intensity of staining in individual cells is probably pretty comparable, suggesting that there's a very robust activation of this pathway, at least in a subset of the retinal ganglion cells at any given time. We can also look within the optic nerve at some of the upstream members of this pathway. So in this Western blot, what we've done is alternate the control mice with the demyelinated delta SOX10 mouse line. And you can see if we stain for the myelin protein MOG, there's a complete loss of the myelin in each of the SOX10 samples, confirming sort of in each case we've got full demyelination. If we look at the expression level of the DLK protein, there doesn't seem to be sort of a consistent shift in the demyelinated versus the control mice. But what you might be able to appreciate is that there's potentially a sort of a change in the apparent molecular weight, such that the demyelinated mice show a sort of a higher molecular weight band. And this might be consistent with either a phosphorylation or a palmitylation post-translational modification in response to demyelination. We can also look downstream, and if we look at, for instance, phosphorylation of MKK4, there seems to be an increase in the demyelinated mice. And we probably also see both an increase in the total density and also the phosphorylation of JNK in the demyelinated mice. So we seem to be seeing a sort of a full activation of the pathway in the optic nerves of these demyelinated mice. Okay, so it's been activated. Does it actually matter? So to try and get a feel for this and whether DLK is actually the initiating sensor of axonal damage in response to demyelination, we made use of some inhibitors that have been commercially available. And it turns out that several companies, including Genentech or more recently Roche, have programs targeting or developing DLK inhibitors for a range of diseases, most notably ALS and Alzheimer's. So we made use of one of these inhibitors, GNE3511, which is an orally available inhibitor of DLK that we can give the mice by gavage. And what we found is if we take these demyelinated MyrfSOX10 mice and give at 10 weeks post-tamoxifen and give them just 6 hours of treatment with this inhibitor of DLK, we can pretty much completely block the activation of c-Jun in the retina. So you can see in these top panels, we've got, again, staining for phospho-c-Jun in the vehicle-treated animals and sort of a sea of activated c-Jun and RGCs. And this is pretty much completely lost in the GNE351 treated retina. This is quantified here. So probably more importantly, obviously, is does inhibiting this pathway actually protect the neurons against death? So we've looked at this once again using the cleaved caspase 3 as a readout of death. Once again, if we take one of these SOX10 conditional knockout mice at 10 weeks post-tamoxifen and stain for cleaved caspase 3 and RBPMS, we can see examples of the dying RGCs right throughout the retina. Here's an example of one of these blowing up here. Whereas if we treat these mice with GNE3511 for 3 days, we get a pretty much complete loss of these apoptotic RGCs. And this is quantified here. So you can see the Cre-negative controls show very little RGC death. We see RGC death in the demyelinated mice with vehicle treatment, and this is reduced almost to baseline levels with 3 days of GNE351 inhibitor treatment. So, we're really very excited about this. I think this pathway seems to be a potentially quite powerful neuroprotective target for demyelinated neurons. So in terms of sort of a working model, we know that DLK is activated in response to a range of axonal injuries and will then initiate a downstream kinase pathway ending in phosphorylation of c-Jun and, in the case of CNS neurons, neurodegeneration. I think what we've shown is that chronic demyelination actually serves as a very similar initiation, somehow activating DLK to initiate this degeneration pathway. So that really has sort of a number of different areas of interest and ongoing questions at the moment. Obviously, we want to know whether the pathway is activated more broadly in demyelinated neurons. We've just started looking at this in the brain and the spinal cord, and we're certainly seeing activation in other neuronal populations. And we're also now trying to extend this into MS pathology tissue to see whether we can also find evidence of activation of this pathway in people with MS. Obviously, we're also very interested in trying to sort of understand the black box, so to speak, at the molecular level between chronic demyelination and DLK activation. Because we think if we can understand the molecular events in demyelinated axons that activate DLK, this might also give us sort of therapeutic targets for intervention. And finally, we're also interested in whether blocking DLK signaling in the long term will prevent neurodegeneration over sort of a more prolonged period. And we're currently following this up with CRISPR-Cas9 strategies to inactivate DLK and other pathway members within the retinal ganglion cells. So hopefully these are all sort of stories I can sort of present at future meetings down the track. So in the interest of leaving some time for questions, I'm going to finish up there, other than to really thank the people that are involved in this work, most notably Greg Duncan, Samantha Ingram, and Matthias Koenig, who really piloted these experiments in our lab many years ago. We've also got a very large number of collaborators, both at OHSU, Baylor College, UCSF, UCL, and Cambridge, many of whom have contributed to this project in other data that I haven't had time to present today. And with that, I'd very happily stop for questions. Thank you.
Thank you, Ben.